Hey, it's Joel Duff, and I found a couple new participants for the Hemoglobin Challenge. Hence, Hemoglobin Challenge episode number 7. Today's participants are Dr. Georgia Purdom and Ken Ham from Answers in Genesis, who managed to team up to misunderstand soft tissue preservation. So, we've got Hemoglobin Challenge episode number 7 coming right up. Before we get to talking about Ken Ham and Georgia Purdom and how they've gone astray and their descriptions of soft tissue preservation, I have to remind you what the Hemoglobin Challenge is. Now, the Hemoglobin Challenge is generally a challenge to anyone who talks about the preservation of soft tissue in the fossil record and doesn't report the data accurately or gives a misimpression to their audience of what has actually been found. And it stems mostly from this idea of hemoglobin that many, well, specifically young earth creationists, often you will use the word hemoglobin as they talk about blood being preserved in the fossil record. They'll say that hemoglobin has been found in the fossil record. And in fact, hemoglobin has not been found in the deep fossil record. So we're talking about like dinosaur bones. Has blood been found in dinosaur bones? Well, something like blood cells have been preserved in dinosaur bones in the fossil record. That's not the same thing as saying the actual cells, but the remains or the preserved remains, altered remains of those particular cells have been found. So, for example, the actual molecule hemoglobin itself is a very complex, large macromolecule, meaning it's a large protein consisting of multiple different parts and thousands of atoms, right? 11,000 atoms total. And embedded in those proteins are 4 heme molecules. And so if a creationist or anybody says that hemoglobin's been found in dinosaur bones, that's not what's been found. That's not accurately reporting the evidence. It gives the impression that this really complicated molecule, right, this really fragile molecule, in a sense, has been preserved for 65 million years, and therefore the audience says, oh, well, of course that can't happen, so the world can't be 65 million years old because how could there be hemoglobin present? But there isn't hemoglobin present. What is found inside of dinosaur bones and remains of other fossils that have been found as well? When we refer to hemoglobin having been found, what really properly should be said is the evidence that hemoglobin was once present has been found. In other words, they've left a trace of their presence, and therefore we can say that there was blood at this particular site in this particular bone. Not shocking, really, that there would have been blood there, but nonetheless, there is a biomolecule that is still present in the bones that represents the breakdown remains of the hemoglobin. So hemoglobin is this complex molecule, 1,100 atoms, most of which is protein, amino acids. And embedded in that are 4 heme molecules, and here's the structure of the heme molecule. And after some time of decay, all of the protein is gone. And what's left is the heme molecules, which are much more stable, being cyclic, polycyclic, aromatic compounds. But not even heme is left in dinosaur bones, right? What's present is the breakdown product or altered remains of what was once heme. So this other figure down here shows, in this case, here's a porphyrin, which is the more generic word for this cyclic aromatic compound that results from, well, it has this nitrogen in it. And so it has carbon and it's hydrogen, so it is a biomolecule. And the carbon and the hydrogen actually is the carbon and the hydrogen that's left from the original biomolecule, the original hemoglobin. And so it is true that the dinosaur itself, like a component of its biomolecules, which we would generically call its soft tissue, right? Because this isn't a hard part of the dinosaur, right? The hemoglobin is part of the tissue, in a way, of that organism, and its molecules, some of its molecules still remain, but they remain in this altered state of a porphyrin. So you have the side chains have been knocked off the heme, and you just have this central core, which is extremely stable, and not surprising that it would persist for tens of millions of years in that particular state. So that was a long-winded way of saying that the hemoglobin challenge is a challenge to young Earth creationists not to mislead their audience by saying that soft tissues and biomolecules like hemoglobin have been found in dinosaur bones. They need to say the breakdown remains, or the very stable breakdown components of hemoglobin have been found in dinosaur bones, which leads us to believe that there was hemoglobin present at one time in those bones. All right, we've got to move on. We got to get to the story, right? So how have Dr. Purdom and Ken Ham managed to mangle soft tissue preservation again? Here's their article from just a few days ago, actually yesterday. Dinosaur soft tissue. Have evolutionists solved the problem? All right, so once again, they're priming the audience. There's a problem here. Dinosaurs can't have soft tissue present because all that should be gone. It can't be preserved over millions of years. So there's a problem. Have they solved the problem? I myself don't think there really is a problem yet, because of what's actually been preserved is not really problematic, right? If there was red blood cells and hemoglobin in their original state, and that's what had been found in fossils, I would admit that would be a curious situation. That would be an interesting problem that would need to be investigated. All right, so here's the article on Ken Ham's blog, and they discussed it on one of their YouTube channel live videos. So let me highlight some of the wording from the article from Ken Ham's blog. In 2004, Mary Schweitzer discovered soft tissue in the femur of a T-Rex. Certainly not the first evidence of soft tissue in the fossil record, all right? But for dinosaur bones, the first time that representations of or remnants of actual cells had been found, right? And so what do they call this soft tissue? Soft tissue is red blood cells, blood vessels, and collagen, right? Now, did they find red blood cells? Mary Schweitzer certainly found things that have the appearance of a red blood cell, but they aren't necessarily the actual red blood cells in the sense that they are in the same state that they were when the dinosaur died. They still maintain the same overall structure of a red blood cell, but the molecules themselves are highly altered from what they originally were. And they're highly altered to very stable molecules, which now persist to the present. How could soft tissue last that long? Well, the soft tissue didn't actually last that long. That's the thing. It hasn't lasted that long. It's been severely affected by time and chemical change. But those chemical changes still maintain a component of the original biomolecules. And the biomolecules have been linked together in such a state that they still represent overall shapes and structures of how those biomolecules were organized in the original cells. And therefore, we see essentially what is the outline of those cells that have been present long ago. Well, evolution has been searching for an answer to this. Again, it's not necessarily a significant problem. It is a really interesting question about how these biomolecules are preserved. I'll admit that, and that's something that we didn't have an answer for 20 years ago. And we're still looking for some answers. I think there's some good hypotheses now. Have they finally discovered the answer? And then they refer to a recent Popular Science article, once again, going to the Popular Science rather than the original research article. I've read the original research article that's associated with that Popular Science article. I'm going to show you that original science article. I don't think Georgia Purdom looked at the original science article or the original research article. She may have, but I think her comments are based just on this Popular Science, very simplified version of that article. The big question that evolutionists must answer is how soft tissue in dinosaur bones could be preserved for millions of years. Once again, it's the right word there is preserved, but it doesn't mean preserved in its original state. It means preserved in its altered, more stable state. Next sentence. Biomolecules, like proteins, are not estimated to last more than 1 million years. And in my opinion as a geneticist, I forgot, we're reading actually Georgia Purdom's analysis now that Ken Ham is quoting in his blog post. In my opinion as a geneticist, Dr. Purdom has a genetics degree, who has worked with proteins in the lab, this length is highly unlikely. All right. I would say all the proteins that Georgia Purdom has worked with in the lab, and I can imagine the types of proteins she worked with, knowing the type of work that she's done. Yes, they're not very stable. And on the other hand, but all the proteins she has worked with in the lab, in her lab, none of those proteins, as far as I know, have been found in dinosaur bones or anything that's 65 million years old, other than collagen. And collagen's a kind of a special case because of its properties already. It is already a very stable molecule, a very stable protein. It becomes more stable when you cross-link the individual protein molecules together. However, soft tissue is found in dinosaurs ranging from 65 to 125 million years. Wow. So proteins can't live for about a million years. I agree. The vast majority of proteins wouldn't survive a million years. They wouldn't survive 100 years under most conditions, or even 5 years under most conditions. Hemoglobin doesn't survive for very long under normal conditions. And it doesn't survive for 65 million years ago. 65 million years. Thus far, I don't know anyone that's found hemoglobin that's 65 million years old. So she goes on to talk about how Mary Schweitzer has postulated that iron is helping to preserve these molecules by reacting with them and forming cross-links. And then there's another new hypothesis now that comes alongside. And the particular paper we're going to look at actually tries to join the two different hypotheses and says these are actually two parts of a preservation process that work together. All right. And lastly, this is a short blog post. Evolutionists are desperate to find a way to make evidence of soft tissue fit with millions of years. I don't see a lot of desperation out there, and they're not forcing it to fit with millions of years. I think it's a really interesting question, which molecules can be preserved for millions of years and which ones don't get preserved for millions of years. What Georgia Purdom and Ken Ham aren't telling their audience is that it's a very select set of molecules that has been found to be biomolecules that have been preserved in these dinosaur bones and any other types of fossils for 100 million years. It's a very specific set of particular types of biomolecules. Biomolecules which any chemist knows are extremely stable molecules, right? The unstable chemical bonds of most other biomolecules are long gone in dinosaur bones. So we know the answer. The simple answer is dinosaurs were buried quickly and deeply in result of the Noah's flood just 4,000 years ago. And so this tissue is well preserved because it's relatively young. All right, I'll flip that around and say if the tissue is relatively young and only 4,000 years old, I could make a prediction, right? The prediction is you should find thousands of different kinds of biomolecules, and you should find them in more like their original state. They shouldn't be highly modified molecules that have been reorganized and linked together in new ways to form these extremely stable molecules that wouldn't necessarily need to have happened to preserve them for just 4,000 years under these conditions that she's describing. And so the presence of specific biomolecules in ancient fossils and the lack of 99% of other types of biomolecules is evidence against exactly the type of preservation that she's mentioning. So no, evolutionists haven't solved the problem, Ken Ham says. The question still remains, how could soft tissue last for millions of years? The answer, it can't. I agree. Soft tissue in its original state can't survive for millions of years. But some aspects of the original soft tissue, the original biomolecules, the original structures that biomolecules make are preserved for millions of years in these highly modified states. Okay, so what Ken Ham and Georgia Purdom are doing is they're setting before their audience, all right, followers of young earth creationism, a false set of both reports of what's actually been found in terms of the descriptions from the fossil record, and then setting up a false scenario where that biomolecules no proteins can survive that long, when that's not anything that scientists say. They don't say most proteins can't survive that long. They don't say that no proteins can survive that long. But it's better for me to use somebody else's words than to try to stumble over my own words. So let's go to the paper. Alrighty, here we are. So the paper that Georgia Purdom is referring to and Ken Ham referring to is from Earth Science Reviews. This paper just published in 2023 by Landon Anderson, and it's as it says, it's a review paper. He's sorted through the literature and is sort of trying to sum up like the current state of knowledge in this particular field. And so he's interested particularly in a chemical framework for the preservation of fossil vertebrate cells and soft tissues. All right, and so what you should know is Landon Anderson is a graduate of the lab of Mary Schweitzer, so he's very familiar with that particular work. And let's just skip down to the introduction. I'm going to read a good section of this introduction. Not going to take very long, but I think it's important for you to hear this general summary of the state of soft tissue preservation. A growing number of studies have reported that recovery of cells and other soft tissues, and you see how he's put that in quotes there, cells and soft tissues, right? Kind of like, things we call cells and things we call soft tissues, but they're not exactly equivalent. It's not like, hey, I've got soft tissue here, and it's not like dinosaurs are like you're opening up that dinosaur bone and you see the same kind of tissue. You see something that represents the soft tissue that was there, and that thing that represents it is a little bit of the actual original atoms that were present in that soft tissue, but they're not in the same state they originally were. Sorry, I'm beating that idea to death, but it's just really important to continue to remember that. These reported soft tissues initially generated controversy within the field of vertebrate paleontology. Yeah, it was surprising at first. No one had really looked for those things. We didn't necessarily have the technology to be able to look and work on that type of thing, and nobody had really thought to look for it, just kind of assuming that it must be really hard to preserve some kind of remain of soft tissue. The first time that was found, yes, it raised quite a buzz, and there was skepticism. And part of that was we didn't really know what that soft tissue is made of. And also it gets reported as blood cells, soft tissue. And so, other scientists are prone to the same kind of misconceptions, right? That they hear those words and they jump to, like the actual remains of an entire cell, like the plasma membrane with the bilipid layer and all these are very fragile things that it's hard to imagine could survive for 50 million years. I agree. I can't imagine the bilipid layer being preserved in its present state in a cell for that long as part of the exterior of a red blood cell. But in fact, those molecules aren't preserved. We don't have those molecules anymore. They are not there. What is there are some of the molecules that have been cross-linked together to hold the framework of what still looks like now the cell, and actually represents where that cell was. Okay, at first, there were challenges. Maybe these are biofilms. Maybe there's another way to explain what things that look like cells or look like nerves or look like blood vessels, but they're really where other organisms have created a skin over the original blood vessel, and that's what remains. Later studies disputed such claims. Yes, I think that those claims are pretty much been laid to rest. I'm sure there's still some believers in the biofilm hypothesis, but that hypothesis has been tested pretty well and is found to be lacking. And there's some experimental evidence in support of these soft tissues being endogenous. However, the notion that cells and soft tissues are unlikely to preserve within mineral vertebrate remains is questionable on its own. I've been saying that since the very beginning, right? This actual claim that they're unlikely to be preserved, what Georgia Purdom and Ken Ham and other creationists harp on, but also other scientists, just in their minds are like, well, it seems unlikely that they're going to be preserved, right? Questioning their likelihood to be preserved. That seems to be questionable. And why? And this is another thing I've said many times, and I'm really glad to see it in print here. Because I always thought this was an intuitive answer. I'm just surprised people who are supporting soft tissues haven't really brought this up as much. Look at this next sentence here. The fossil fuels used daily by society consist of original biomolecules, right? They're the original biomolecules, the original carbons and hydrogens and nitrogens and oxygen that was in the molecules of organisms at some time, right? Those fossil fuels that you're using, of ancient plants and microorganisms that have been chemically transformed into carbonaceous macromolecules referred to as kerogens, right? Oils are not in the exact same state that they were in those original organisms, in those plants and microorganisms, right? They've been altered. They've been chemically transformed. So they've broken bonds and reshifted the bonds around to form these carbonaceous macromolecules, which we call kerogens. Many of which now are going to be these cyclical aromatic compounds, which are highly stable. In cases such as coalified fossil wood, for example, the conversion of biomolecules toward kerogen molecules can preserve original tissue morphology. Yes, we see that in plants. See, Ken Ham, George Purdom get really excited about these organic molecules or biomolecules found in dinosaur bones, but there are plants that are just as old in the fossil record that have, you can see the outlines of the cells, right? You can see internal structures of the cells, right? Those original biomolecules have been turned into these kerogen macromolecules, which maintain the shapes of the original cells, right? They preserve the original tissue morphology or shapes and characteristics. The field of soil and petroleum science even accepts, probably my head's on top of that. Let me scroll down here. The field of soil and petroleum science even accept the recalcitrant biomarkers can preserve through time as portions of these highly cross-linked kerogen macromolecules, right? Petroleum scientists have long accepted that biomolecules can survive for hundreds of millions of years. Further, the preservation of biological tissues is a phenomenon known to occur within invertebrate fossils, right? Insects. For example, pre-Pleistocene arthropod cuticles have been experimentally shown to consist of hydrocarbons, carbon and hydrogen linked together, which is very similar to the original biomolecule in that cuticle. And so it's not been replaced by another hydrocarbon, it has just been altered, right? The original biomolecules have been altered, and they still just maintain the same shape and structure, some of the same structures that they originally were in those insects. Such findings support, let me scroll down here. Such findings support that the original cuticle is still present in some form and has not been fully replaced by exogenous materials, just meaning haven't replaced the biomolecules with some other, mineral, right? So the biomolecules are there. That's soft tissue preservation. That's what soft tissue preservation is. You're preserving the original biomolecules. Multiple publications have hypothesized that such preservation is a result of cross-linking chemical transformations of original cuticle chitin and proteins. That's not really any different than what Mary Schweitzer and I think it's Weisselberger-Weisman are going to be suggesting for how soft tissues, cells, and other macromolecules have been preserved in altered states through time in dinosaur bones. Indeed, two mechanisms have been proposed to explain reports of soft tissue and biomolecules within ancient vertebrates. Both result in the intra- and intermolecular cross-linking and chemical transformation of reactant biomolecules, a process referred to as in situ polymerization. All right, so intra means within molecules, so that's, say, a heme molecule. And if bonds are changed such that a bond shifts and connects to a different molecule within that heme molecule, that's an intramolecular, intermolecule cross-linking. I'm sorry, actually, if you even bind one heme molecule to another, I think that's intra cross-linking because it's the same type of molecule. And then you can have one molecule linked with another molecule, right, a different kind of molecule, and bind those together. So you can get long scaffolds of proteins that are all bound together that in the original material, right, in the original organism, weren't bound together. It's kind of like saying I've got 100,000 different proteins in my cells, and they're all separate from each other, doing jobs. But under the right conditions, many of them would link together, right? You can get them to stick together. And if you had them chemically transformed such they stuck together, you'd form a very large macromolecular complex. And that sticking together can actually add stability to those molecules. Okay, Now, we're almost done, actually. Right? What I'm going to do here is I'm going to just scroll down. I want to show you some images because I've been emphasizing that what's preserved in soft tissues are particular molecules that are more stable than other forms of molecules that might have been found in the original soft tissue, right? The original biomolecules. So here are kerogens, and here you've got those cyclical aromatic compounds, right? So these are carbons all around with a few hydrogens. And let's just keep going. Here's a bunch of oil. All right, you've got carbons and hydrogens, and you have a process which you begin with a biomolecule in a living organism, right? And it has phosphate groups and nitrogens and so forth. But as you chemically transform it and you cross-link it together, so here you have separate hydrocarbon chains. But ultimately what happens is the hydrocarbon chains will get linked together to form what? Cyclical aromatic compounds, which are far more stable. Right? They're not going to just fall apart and break apart easily. Let's keep on going. Lots of chemical formulas in here. Here we have tyrosine, which is an amino acid residue that's part of proteins. And proteins, through changes, interaction with water and so forth, you can get the tyrosines to cross-link together. And you get more and more and more cross-linking, which then eventually results in the formation of cyclical compounds, which again are more stable than the original ones. And what you have is a loss of side chains. Just like in the heme molecule, you lose the side chains, so you just are left with a cyclical compound. Right? So what is not stable, what is more prone to decay, right? Chemical decay, is lost over time. And there's been a lot of time, right? And what remains are these really stable molecules, which at some point, chemists can tell you that some of these compounds are, as long as they're not going to be introduced to, say, bacteria or something that can attack them and eat those and use them as a carbon source, right? If they're preserved from that in the environment, there's not a lot of things that's going to break that down at some point. So once you have a stable amount, the temperature and the amount of water in the environment is limited, you then don't have any reason to believe that these won't break down, right? That you don't have any reason to believe they will break down, right? You would expect that they should last for millions and millions and millions of years. Unlike what Georgia Purdom tells her audience, which is, oh, well, I'm a geneticist, I work in a lab, and I've seen proteins, and they don't last for a million years. They don't even last for hundreds of years or tens of years in my lab. But those aren't these molecules, right? She hasn't worked with these molecules. Right? She can't apply her observations of a few molecules to all molecules. We're talking a very small subset of overall molecules are being preserved. Okay, let's just keep on going. I know there's a couple more in here. You get the idea. We're talking about a lot of different, actually there wasn't any more. But the whole point is that cross-linking between molecules and within molecules eventually results in a greater preservation of those molecules. And since you're cross-linking between molecules, you're putting things together that before in a living organism were separate, and you're attaching them together. That's how you maintain the overall shape and form of some of the original structures, right? Collagen also is a very long molecule. It's a very long protein, and you get lots of cross-linking between it, like all those tyrosine molecules, and you get cyclical aromatic compounds embedded in there, and then you embed that inside of bone, right, that's being preserved and encased, and you're going to have preservation of collagen for a long period of time. But again, that collagen is not like the same as the collagen that was in the organism right when it died. It's been changed greatly. It's been dramatically altered. So I think that's it for episode number 7. All right, it's pretty simple. Ken Ham, Georgia Purdom are under the misconception that soft tissues somehow are the remains of the original material. And I'm going to say that carefully again. They are the remains of the original material in the sense that there are atoms and carbon bonded molecules that are biomolecules that are still present that represent atoms from those original cells, but they're not all bound in the same way anymore. They've been reconfigured over time, and that's what lends to their stability. So they're absolutely right that molecules like hemoglobin, if I were to just take it out of your blood cells right now, and I were to try to preserve that in some way, in the sense that it won't break down into pieces, right? It won't fall apart into submolecules, and then eventually the bonds break apart. If I were to try to preserve it in its original state, it'd be extraordinarily difficult, and it'd be really hard to believe that that could survive for 65 million years under any conditions. Although the caveat always is like, we don't know all the different conditions of preservation. Maybe there's some exceptional condition in which hemoglobin in its larger sense could be preserved over longer periods of time. I don't know what that would be, but I'm not going to deny the possibility of that happening. I do think it's unlikely. But the current state of the science, which is what I'm interested in Georgia Purdom, Ken Ham, and other creationists working with, is that we haven't found hemoglobin, right? We haven't found collagen in its original state in fossils. And they need to tell their audience that, to be fair, to be true to their audience, and let them know what the actual state of the research is. All right, that's it for today. Hemoglobin Challenge episode number 7. Hopefully, I'll have some. Remember, I'm going to do positive challenges as well. I mean, I'm going to, not challenges. I will do episodes in which I congratulate people for properly identifying the nature of soft tissue preservation. Unfortunately, it's only happened once so far, right? Out of the 7 episodes now, only one of my episodes has been like, great news. Somebody has actually approached this in the right way and changed the way that they have said it. So that's my goal. Have Ken Ham and Georgia Purdom, the next time they talk about this, not say the same thing, but be more careful in their language. They can still try to spin it, to reinterpret the data to somehow fit with a young Earth, but they at least have to be honest with what's been found and reporting the knowledge that has thus far been discovered. All right, that's it for me. Thanks, I'm Joel Duff. Like, subscribe, do all that good stuff, and we'll talk to you later. Bye-bye.